Hi students, welcome to topic 2. This is focused on microscopy, staining and safety while working in the microbiology lab. There's two parts to this topic, part 1 and part 2. This is part 1. I'm showing the intended learning outcomes for topic 2. So topic 2, you're expected to list the rules and regulation while working in the microbiology lab. Okay, you're demonstrating awareness of the lab safety in the while working in the lab. Okay, to describe the various type of microscopes, we'll look into that later, as well as the function of the various components of a light microscope. Okay, you'll be asked to explain the principles of staining techniques, and as well as to do and perform some of the techniques during the practicals. Okay, so let's dive into the first point here. For rules and regulations in a microbiology lab, laboratory safety is of importance. So the laboratories in the food and the biomedical sector, as well as in the chemical and pharmaceutical industries, each have their own safety regulations and safe working practices, as well as their procedures. So when you are heading out for your internship, you can expect their own safety regulations that will be communicated to you. So the laboratory safeties are also a set of rules and regulation written, usually in a safety manual. Okay, so as to protect you as well as your colleagues and your people working in the laboratory. And often you will need to adhere to them strictly to avoid any injuries, and hopefully we don't have any death around arising from this. Okay, I'm outlining some of the rules here. The first rule is to always wear your PPE, your personal protective equipment while working in the lab. Okay, this includes your laboratory coat. Okay, wearing long pants also help to prevent any spillages from coming in contact with your skin on your thighs. Okay, wearing covered shoes is important. Okay, just to protect your toes if something were to land on your toes, as well as to protect from any contact, be it a liquid or solid coming into contact with your legs or toes. Okay, eye goggles are important when you're doing any open bench experiments, as well as the wearing of gloves. You will notice that there are two types of gloves and even more types of gloves in the lab. Okay, these are your nitride gloves on the left here. Okay, those are in blue. They have good chemical resistance. All right, and it's an overall good choice, especially for people who have allergy towards latex. Latex gloves are on the right here. Okay, you will find this latex gloves are typically being used in the hospital setting. They are good barriers against blood, but they have poor chemical resistance. If you're using chemicals, preferably nitrile gloves will be the way to go, and especially if you have any latex allergies. So rule number 2 is to familiarize the safety equipment in the laboratory. Yeah, so it's important that users be familiarized on the usage as well as the location of this safety equipment. This includes the eye wash, the first aid kit, the emergency shower, okay, fire extinguisher, as well as the exit sign. Yeah? Okay, the eye wash is important because in case your eyes get in contact with any chemicals or any solution, okay, it's important that you run to the location where the eye wash is located. Okay, typically this will be next to a sink, and then the individual will have to flush his or her eyes, okay, for a good at least 15 minutes, just to get rid of any solution that have went into the eye. Alright, first aid kit contains your necessary materials, bandages, safety pins, as well as your plasters, so you can cover up any of your open wound with the materials here from the first aid kit. Okay, emergency shower is also important. Okay, know the location of where they are while working in the laboratory. Okay, they are useful, especially you have large amount or even small quantities of corrosive chemicals that got into your body. Okay, it's important that you walk briskly to the shower, and then just take off your lab coat, and then start to flush away any of the contact of the chemical or solution. All right, for fire extinguisher, know the location. These are typically good for electrical fire, or even for chemical fire arising from its usage. All right, and finally the exit sign. Okay, typically the entrance will be the exit, but do take note of this signages so that in the event of emergency you will know where to evacuate. Okay, rule number 3 is to adhere to all established safety regulations in the laboratory. Okay, you will want to tie up long hair if you have any, and then restrain any loose clothing. So, for example, if you're wearing a tie, this should be taken off, just in case the hair or the tie get caught in the equipment. There should be no drinking, eating, smoking, or applying cosmetics, or even vaping in the laboratory. Okay, if you have any open wound in the skin, please do cover it up with a plaster before working. Okay, label all materials, be it chemical or specimen clearly with a marker, so that we will know what the container contains. Okay, you will come to across many of the chemicals to be colorless. You won't even know what is the chemical, so it's important to label them accordingly. So report all incidents to the staff in charge of the laboratory. This could be your supervisor, or it could be your lecturer or tutor in the laboratory. Okay, before you leave the lab, okay, it's a good practice to wash your hands with antiseptic soap, and then never work alone. Always work with a buddy or someone in the lab. Okay, always assume that all microorganisms are capable of causing diseases. Don't take your gut down. You never know. In the case of microorganisms, it may be harmless, but if you manage to get into your body, okay, remember from topic 1, okay, they may be harmless, but they may become opportunistic pathogens. Okay, practice aseptic techniques during manipulation of microorganisms. Okay, the aim here is to minimize the aerosol formation. Aerosol formation means the production of gases or bubbles that can lead to the spread of microorganisms in the air, and also effectively causing cross-contamination between your specimens. So aseptic techniques will be taught during your practicals. Okay, this includes probably flaming of your inoculating loop, and then opening your specimens in the smallest way possible, okay, so as to minimize cross-contamination. And this also includes working near a Bunsen burner flame. Okay, more details are in your laboratory manual, okay, as well as during the practical session. While working in the microbiology lab, it's important that you recognize the GHS pictograms. GHS stands for the Globalized Harmonized System. So these are hazards which have been harmonized across countries internationally, and these are the pictograms that signify to the user certain hazards that needs to be taken note of. So this includes the pictograms such as this that denotes serious health hazards. Pictograms such as this with flame, meaning that the chemical or the reagent may be flammable, and so on and so forth. So you will need to know the pictograms here, and then how it relates to the hazards that comes with it. So as we work in the lab, inadvertently you will generate waste. So it's important to follow proper waste management and disposal procedure. Okay, before starting work and before and after, do wipe down and disinfect your work benches and surface with 70% ethanol. All right, 70% ethanol, all right, with an exposure of more than 5 minutes is bactericidal. When I mean bactericidal means that it's killing the bacteria. Okay, however, it's not effective for bacterial spores. Okay, we'll talk more about the spores in later chapters, but for now, just know that in spore states, the bacterial spores are actually vegetative cells, means that they go into hibernation, they don't grow, they don't propagate, they're just in a so-called sleeping status, and they're quite resistant towards 70% ethanol, even at more than 5 minutes. Any spill bacterial culture or microorganism culture, especially in large volume, they should be cleaned up with an appropriate spill kits. So this could be your biological spill kits, okay, if they are bacterial in culture. So the biological spill kits that we have in the lab looks like this on the right. Okay, if you have a chemical that mix together with the cultures or they are chemical in nature, then the chemical spill kits needs to be used. All right, if in any case, do notify your tutor or your lecturer or your supervisor in the case if you're heading out for your internship, right? There's a spillage, so that the cleanup and management of it can be done properly. Okay, all microbial contaminated material shall be disinfected with sodium hypochlorite. Sodium hypochlorite is also known as a household bleach. Okay, once they are disinfected, okay, usually this is done overnight for a certain period of time, more than 24 hours. They are subsequently autoclaved in yellow biohazard bags shown here. Okay, the bins that you see here on the left hand side, all right, these are the bins that you will find them in our laboratory. here ASC. So often we will discard the materials, the contaminated materials in double bag yellow biohazard bags, okay, and subjecting them to autoclave. Right? If you have any sharps that you'll be using, maybe inoculation needles or tips. All right, this needs to be disposed of properly, especially the sharps that contains blades. All right, they should be disposed in puncture-proof yellow sharp bins for incineration, as shown in the photo here. These are sharp bins that contains biohazard materials. All right. So most of the microbes cannot be seen by the naked eye, except for microbes that we can see with our naked eye. We term them as macroscopic, M-A-C-R-O. Macro means the bigger scales of things. So here, for example, are the mushrooms, are fungi from the group of fungi, or the kelps we see from the group of algae. So to study microbes that are either unicellular or multicellular, they are microscopic in nature. We will use typically a light microscope. Right, for viruses, they are much smaller than bacteria and eukaryotic cell. We will need more sophisticated microscope known as the electron microscope. So here I've outlined in the table here some of the microorganisms that we talked about earlier and some of the average sizes. You will see that the protozoa is much bigger, okay, compared to the yeast and bacteria, and then viruses are much smaller in sizes relative to the rest. Okay, just to recall back to topic 1, right? Viruses are often acellular, whereas the rest of the microorganisms here, bacteria, yeast, and protozoa, they are cellular in nature. So let's switch gear and then talk more about the light microscope. The compound light microscope, also known as a light microscope, some of them call it as a bright field light microscope. It's all the same, but generally you will see different components. One of the components will be the ocular lens. This is where the user can view the specimen that's been mounted on the stage here together with the objective lens so that you can provide a magnified and large image of the specimen. So the two lenses here, the ocular and the objective lens, are taken into account when you want to calculate the total magnification. So typically the ocular lens are 10 times in magnification, right? The objective lens are available in different flavors, okay? It can be in 10 times, 4 times, 20 times, 40 times, or up to 100 times. All right, so if you want to calculate or determine the total magnification, you should take the ocular lens magnification together with the objective lens magnification. So this will be 100 times 10. This will be 1000 magnification. All right. The ocular lens where the light will be focused on. Okay, coming from the light source here at the bottom, indicated by the sun logo here. Okay, passing through the condenser, and then this is where your specimen sits at the specimen stage. Okay, and then this will be magnified through the objective lens, and finally image will be formed on the ocular lens. All right, so take note that the capital big X after the different magnification index on the objective lens. So the objective lens, as I mentioned, will magnify the specimen on the stage. All right, different flavors, 10 times, 4 times, 100 times. Okay, do note that the 100 times objective lens will need oil immersion. These are typically immersion oil that help to maintain the reflective index of the light when light passes through the objective lens. The light pathway, as I mentioned just now, all right, this will pass through the condenser and then focuses on the specimen on the stage. So the image here on the right hand side shows you different objective lens. The red one is in 4 times. The yellow objective lens could be a 10 times or 20 times. The blue labeled objective lens are typically 40 times, and then the black or the white labeled objective lens are 100 times. When we talk about light microscope magnification, we also talk about image generation. Right? Often resolution will come into the picture. So resolution refers to the capacity of the light microscope, or in fact any microscope, to distinguish 2 points or 2 objects that are next to each other. All right, and how we provide that magnification and resolution is to use the coarse and the fine adjustment knots on the microscope to differentiate between these 2 points. So the coarse adjustment knot is the big bulky knot here, as you see, right? The fine one, which is circled in green here, these are the fine adjustment knots. Typically, when we work through an image or to magnify an image, we will start with the lowest magnification. So this is often using the 4 times objective lens, and then moving up your way up 10 times, 20 times, okay, using the coarse adjustment knot. Once you're at 20 times magnification, there's no need to use the coarse adjustment knot anymore, and often we will just need to focus using the fine adjustment knots as we work our way up from 20 times, 40 times, and finally to 100 times. In addition to resolution, okay, contrast is also important. Okay, specimen contrast is defined as the ability of the detail to stand out against the background of other neighboring details. Okay, contrast is basically dependent on the interaction of your specimen with the light, and this is dictated by this component here called the field diaphragm. You need to increase the field, or some call as iris diaphragm, to increase the visibility of the particles and the edges in the specimen. The field diaphragm is typically found at the bottom of the stage, which is highlighted in yellow here. So the principle of light microscope depends on your visualization of your specimen, especially at 100 times. The highest power that the objective lens can go at 100 times. Often you will need immersion oil. The immersion oil coming in contact with the objective lens as well as the specimen on the glass slide will allow you to achieve a good resolution. This is because the refractive index of the oil is similar to the objective lens. All right, at 1.5. Without the use of oil, as you can see here, the immersion oil in contact with the glass slide specimen as well as the objective lens here. Okay, without using the oil, this will cause the light passing through the specimen to scatter into the air. Right, the air does have a lower reflective index. All right, and hence this will decrease your resolution, making your image blur and you can't see the image clearly. So I'd like to go through some of the different types of light microscope. There are generally 4 types of light microscopes you need to know in this subject. These are the bright field microscope, also known as your light microscope, your dark field microscope, okay, your fluorescent microscope, as well as your phase contrast microscope. So let's take a look at each of these microscope in detail. Okay, so the different types of microscope is application and images. All right, so for the bright field microscope, these are generally suitable for dead, stained specimen or live unstained organisms. So you see here the paramecium in the bright field microscope. You can see that you can see the cilia as well as their vacuoles within the paramecium. This is a live microorganisms, and hence it can be readily visualized using the bright field microscope. Okay, for the dark field microscope, okay, these are suitable for live unstained microbes. No light is going through, hence the word dark field. Okay, it's blocked off already. But the only light is being allowed to go through will be reflected off the side of the specimen itself. So if you're interested, I've included a link here to the image of how a paramecium will look like in the dark field microscope. Yeah, fluorescent microscope, they are suitable for specimens that are stained with fluorescent antibodies. So antibodies can be coupled with a fluorescent. Okay, that fluorescent will fluoresce and give off energy when you expose it to UV or certain lasers. All right. Again, if you're interested, you can compare how they look like between the different microscopes. I've included a link here that will direct you to how a paramecium will look like under a fluorescent microscope. Okay. Phase contrast microscopes, okay, they are suitable for live specimens with observable inner structures such as your granules, your cilia, your organelles, right? So again, I have included a link here if you're interested to take a look of how a paramecium look like under a phase contrast microscope. Okay, there's another type of microscope that gives you the differential interference contrast, also known as DIC. Okay, this DIC microscope is again suitable for live unstained specimen. So it provides you with color and also a 3D image of the specimen. So look at how a paramecium look like under DIC here, the image on the right. Here you can see the vacuoles are green in color. This could be a paramecium that have taken on board some food vacuoles within their intracellularly. All right, and in overall, you can see their surrounding cilia also, but not very clear compared to your bright field microscope. All right, so it's important to know what are the features of these different microscopes, and how are they used to study the structures of microorganisms. So in addition to light microscope, there are also electron microscopes. Okay, electron microscopes are much more specialized and for sophisticated. They allow you to visualize materials, be it viruses or even red blood cells at great detail. Okay, so there are two types of electron microscope. First is the scanning electron microscope, also known as SEM, as well as the transmission electron microscope, also known as TEM. What you will see is that the specimens are often stained with metal, right? And they are bombarded with electrons emitting from this microscope itself, so as to provide an image to the user. In the case of SEM, this will provide a 3D image. Okay, I've included a image here on the right hand side. This is Plasmodium falciparum. Okay, this is a parasite causing malaria, infecting red blood cells here. So you can see they have a lot of bumpy features on the red blood cells and very crumpled looking on the red blood cells surface. These are infected red blood cells with Plasmodium. Okay, coming up to recall back what we learned in topic 1, Plasmodium falciparum, this is the parasite. Plasmodium will be the genus name. Okay, falciparum will be the species name itself. So see how I have written the microorganism name based on the binomial nomenclature here. Okay, in contrast, the TEM, transmission electron microscope, okay, not only the specimens are stained with metal, okay, they are also sectioned into thin slices so that you can increase contrast and have a look at the cross-sectional dimension of the specimen itself. So it helps to reveal detailed structures like the organelles within the cell. So if you could see the image here where my pointer is, this is a similar Plasmodium falciparum parasite infected red blood cells. Okay, the cross section of the red blood cell is being sliced, and then you can see the internal structures within the parasite itself with TEM. So in summary, we have went through the rules and regulation while working in the microbiology lab. Okay, this includes wearing your PPE, that includes a lab coat, having covered shoes and long pants, all right, as well as wearing eye goggles if you're working with any open tube experiments in the lab. Okay, adhering to safety regulations that include tying up your long hair, all right, and then restraining any loose clothing such as a tie. Okay, you don't want your tie to be stuck in any of the equipment that we use in the laboratory. All right, as well as following safety procedures. This includes the waste disposal procedures. All right, disinfecting your benches before and after work with 70% ethanol. Remember, 70% ethanol is a bactericidal agent that kills the bacteria. Okay, however, it's not effective against bacterial spores. All right, proper disposals of biohazard material should be placed inside yellow biohazard bins, but if they are sharps, they go into the sharp bins, right? All right. Importantly, also when you're working in the microbiology lab, you need to familiarize yourself with the safety material, safety equipment. These include the eye wash, for example, or the showers. We also talk about the different types of microscope. Okay, there are 5 types of microscope. Okay, generally these are your bright field microscope, dark field microscope, fluorescent microscope, the phase contrast microscope, as well as the differential interference contrast microscopes. So these different microscopes, we have talked about each of their features and what they are suitable for. It's important that you know which are their features and what they are operating on. Right? We also talk about the different functions and components of a light microscope. This includes the ocular lens, the objective lens, the field diaphragm, the condenser, the stage where you place your specimen, as well as how you can determine the total magnification power of your microscope, taking into account your ocular lens, which are often at 10 times power, okay, together with your objective lens, and this may be from 10 times all the way to 100 times. 100 times magnification, right? So with your objective lens at 100 times magnification, often you will need to use the immersion oil as well, so that you can produce a clear and image for you to visualize your specimens. So this is the end of part 1 for topic 2. I will see you in the next topic. Thank you. Hi students, welcome to part 2 of topic 2. Topic 2 is focused on microscopy, staining, and safety in the microbiology lab. In this part of topic 2 e-lectures, we're going to look into more details about the principles of staining techniques. As we learned earlier in part 1 of this topic, okay, we use the light microscope or even more advanced microscope such as the electron microscope to visualize our specimens. Okay, these specimens may be arising from microorganisms of different sizes. Alright, so the preparation of specimen for microscope is important. Typically, we will mount the specimen or the sample itself on a glass slide. Some factors to consider during preparation or before preparation is, are the specimens living or dead? Okay, because this will determine what type of light microscope, or is it the electron microscope you'll be using. Right, what's the aim of the experiment? Do you want to see the overall structure? Do you want to see motility or movement? Right, and the type of microscope that's available for your use. Right. For fresh living preparations, these are typically done for live microorganisms. They are either placed in a wet mound or in a hanging drop mound. Wet mound are typically done more commonly using a drop of the culture with a cover slip on the glass slide. The less common method will be a hanging drop method. So here I show one of the image here on how a hanging drop is being done. Right, typically it will be a glass slide as well as the cover slip, but you will use some plasticine to encase the whole glass slide, the cover slip. The cover slip often will have a drop of the culture specimen that you intend to visualize, and this will be sealed together with Vaseline. Okay, less common in the industry now in the microbiology lab. For example, we hardly do hanging drop now unless that the specimen is of small volume or difficult to visualize under a wet mount. A more common wet mount will be sufficient for most of the specimen. So if we were to look at fix and stain preparation, this is different from fresh and unstained or unfixed preparation. We want to fix the microorganisms so that we can do more long-term study and also archival of these microorganism specimens. So typically we will do a smear technique. So this involves making a thin smear from a solid or liquid culture of the cells on a glass slide. When I say about solid or liquid culture, okay, we will look more into this as we go down the topics here in this subject. But solid culture, just for knowledge now, right, typically refers to colonies, bacterial colonies that are from a agar plate. Right, bacteria in the liquid broth can be found in liquid culture. Typically, these are in the liquid nutrient broth itself. All right. So once you have make a smear, and we will do this during our practical. All right. We allow it to air dry for about 5-10 minutes. Once it's completely dry, we will do a heat fixation. Okay. Heat fixation kills the specimen and then secure the specimen to the slide. Okay, also aims to preserve the cellular components with minimal distortion to it. Right, heat fixation can be done using heat, using a flame from the Bunsen burner, for example, or sometimes fixation can also be done with alcohol or formalin, okay, chemicals that can kill the specimen and adhere them to the glass slide. Right, similar to a photograph, right, last time we will still need to develop the photograph using stains, right? So similar for your smear, to develop the smear, we perform staining using dyes. So dyes can be classified either basic or acidic. Okay, basic dyes are cationic in nature. They have mostly positive charges. Alright, they can also be acidic in nature and ionic, having negatively charges. Right, so we know that cell components have different charges on them. Some can be positively charged or negatively charged, and hence these different cell components will attract the dyes accordingly and stain readily. Yeah? So for example, if your cell components of a bacteria has a lot of negatively charged acidic substances, all right, this will react with basic dyes, right? They're negatively charged, the basic dyes are positively charged. They will come together, resulting in a positive staining. All right, So note that some of the acidic dyes, okay, have negatively charged, right? They tend to be repelled by cells, and hence they are very good for negative staining. All right, some of these are negatively charged acidic substances you can find on the bacterial surface are like, for example, your lipopolysaccharides, okay, as well as your tachyoic acid that is found in your peptidoglycan cell wall of your bacteria, right? Be it a gram-positive or gram-negative, right? You will notice that the gram-positive bacteria, they have a thicker peptidoglycan, right, compared to the gram-negative bacteria peptidoglycans. Gram-negative bacteria typically have a lipopolysaccharides, and these lipopolysaccharides are actually important for immune recognition by your white blood cells. So when we talk about positive and negative staining, remember it relates to the dyes charges as well as the cell component charges. So for positive staining, okay, cells are typically colored with a white background. So you can see here the positive staining. You can see a lot of bunch of cells, bacterial cells here. They're all colored, right? Different color. You got purple, you got pink, right? The background is generally not stained at all, right? They're generally white. Okay, so here we are looking at some of the basic dyes like crystal violet, like safranin, right? These are your positive staining dyes, basic dyes, right? Negative staining cells are in fact clear, colorless, they're not stained at all, but the background are stained. All right, so in negative staining, okay, you can see here, this is an example also of where the background are stained either dark gray or black, and we use some of these acidic stains dyes such as your Indian ink. All right. When we talk about staining, you have different types of stains, right? A simple stain typically use one dye, right? Whereas a differential stain, such as those that uses two different dyes, for example, in gram staining, right? It helps us to differentiate between different components of the cell itself. So in gram staining, which is the focus of this topic here, all right, it helps to differentiate between gram positive and gram negative. Okay, remember positive will be your cells staining purple, right? These are your gram-positive bacterial cells, right? For gram-negative bacterial cells, these are your cells staining pink or red, right? We'll go through the concept of how this allows the differentiation between gram-positive and gram-negative bacteria in the later slides. But importantly, with gram staining as a differential stains, and hence we use two different types of dyes here. This includes the crystal violet and the safranin dye. It helps to guide treatment for the patient, or even drug selection for patients with sepsis. Sepsis is a systemic bacterial infection in the patient. It's important to recognize what kind of species the patient has or has been infected, and these species may be gram-negative and gram-positive. And this will help the physician to dispense appropriate treatment such as antibiotics for the patient recovery. Okay, another type of differential stain will be your acid-fast stain. This use a fusin dye along with other staining dyes. This is mainly for your information now to detect Mycoplasma tuberculosis, the bacterial species that's very slow growing, but yet very important as it causes tuberculosis, infection in the lungs. So acid-fast stains, one of the component is the fusin dyes. We talk about the simple stain using only one dye, differential stains such as your gram stain and acid-fast stains. These are important and they use the theory of positive staining. Negative staining, you only need to know Indian ink as one of the acidic dyes, and these are typically useful to stain for the bacterial capsule or the spores. So we talked about gram staining just now as a differential stain. Gram staining allows you to differentiate between gram-positive or gram-negative bacteria. Okay, gram-positive bacteria, they stain purple, all right, once you apply the stain on them because they have a thick peptidoglycan, as you can see here on the right-hand side here. The peptidoglycan consists of your tachyoic acid as well as your lipocharic acid. It's a very thick peptidoglycan cell wall here, right? In contrast, the gram-negative bacteria, once you perform a gram stain, they will appear as pink or red because they have a thin peptidoglycan. The thin peptidoglycan, as you can see here, In addition to their thin peptidoglycan, they have also a outer membrane that consists of your lipopolysaccharides, which I mentioned just now. They are important for immune recognition by your immune white blood cells in the body, yeah? As well as other attachment factors. So most bacteria are either gram-positive or gram-negative, be it purple positive or pink as gram-negative. Only a very small group of bacteria are gram-variable. So when I say gram-variable, means that there's a mix of gram-positive and gram-negative phenotype when you look under the light microscope on these stained specimens of bacterial smears. So this image just shows you the difference between gram-positive and gram-negative. So gram-positive bacterial cells, once they are gram-stained, okay, they will appear purple. This is due to their thick peptidoglycan. Okay, they will retain the crystal violet complex here, whereas the gram-negative bacteria, because they have thin peptidoglycan, right, they are not able to retain the crystal violet dye here or the stain here readily. Okay, they will in fact take on an additional dye, which is safranin, and hence they appear pink under the light microscope. So note the difference in the structures of the peptidoglycan between gram-positive and gram-negative. Now I want to talk about the stains that we use during gram staining. The stains are typically the crystal violet stains, all right, the iodine, okay, as well as a chemical as 95% ethanol as a decolorizing agent, as well as safranin as a counter stain. So the gram staining is able to help us to differentiate between gram-positive and gram-negative bacteria. So after heat fixation, typically we'll apply the crystal violet for about a minute. This will stain the cells purple as a primary stain. Okay, after washing it off with water, okay, we'll apply iodine. Iodine acts as a mordant. It helps to fix the primary stain in terms of the crystal violet here by forming a crystal violet dye complex within the peptidoglycan. Okay, in this stage now, both cells will still appear as purple. All right. But after decolorization with 95% ethanol, okay, this dissolves the lipids in the outer cell membrane, especially in the gram-negative bacteria, right? And because of its thin peptidoglycan, okay, the dye complex is being washed away. Yeah? So the thick peptidoglycan will still retain the crystal violet complex here within the cell itself. So hence, this will allow you to differentiate between gram-positive and gram-negative, because positive bacterial cells, gram-positive bacterial cells stains purple. Right, with the decolorized gram-negative bacterial cells, they readily take up the safranin stain as a counter stain, as a secondary stain, and hence they will appear purple under the light microscope. So more details during the practical, you'll be doing gram staining, so keep a lookout for it. So I just want to emphasize the difference in the gram-positive and gram-negative bacteria. Gram-positive bacteria have a thick peptidoglycan, the cell wall. Okay, in contrast, the gram-negative bacteria has a thin layer. All right, the gram-positive bacteria, they retain the primary stain due to the thicker peptidoglycan layer, and hence they will appear as purple when stained with crystal violet. All right, in contrast, the gram-negative bacteria do not retain the primary stain or crystal violet due to their thinner peptidoglycan layer. Okay, the gram-positive bacteria, they do not take up the safranin counter stain. Okay, whereas the gram-negative bacteria, they do take up the stain and hence appear pink, and this is after the step of decolorization using 95% ethanol. Alright, and under the light microscope with the bacterial smear preparations, you have gram-positive bacteria appearing purple. In contrast, the gram-negative bacteria appear pink. Now I want to switch gear and talk about bacterial shapes and arrangement. This is a non-exhaustive list. It's just a sampler of what are common shapes and arrangement we can come to expect from bacteria routinely, commonly. So bacterial cells are prokaryotic. They are unicellular microorganisms. The bacterial morphology refers to the bacterial cells and shapes as well as their arrangement. They may come in different shapes. So shape of a coccus, like cells is spherical or ball shape. For example, the genus of Streptococcus. All right, they look like ball shape, spherical in nature. Okay, you have the rod shape or bacillus-shaped cells, bacterial cells such as E. coli or Bacillus anthrax, or they can come in a curved form, where they could be gently curved or spiral shaped. For example, in the microorganism of Campylobacter jejuni, which is involved in various pathogenic infection in humans. So bacteria can also arrange in different forms. All right, those bacterial cells in coccus or cocci arrangement, they may appear as single and in pairs. We call them diplo, tetrad, group of four, or they could be in irregular clusters or in packets, like for example Staphylococcus aureus that causes skin infection. Okay, they may be happening in nature in chains also, like for example Streptococcus pyrogenes, which are in chains like this, and often they cause sore throat. Okay, for bacterial cells, they are in a bacillus or rod arrangement. They can come in singly form, like Pseudomonas, which are responsible for lung infections. They may come in pair, again we call them diplo, or they may be in chains, just like this. So how do we describe bacterial cell morphology? We must describe in the following format. We first determine the gram status, okay, from the gram staining. We talk about the shape of the bacterium, be it is it coccus or cocci or bacillus, rod shape, and then we also talk about the arrangement. Are they in diplo, tetrads, or clusters, or in chains, or they are singly formed, right? So for example, when you look under a light microscope, you see a phenotype like this. Alright, the bacterial cells are stained purple, hence gram-positive. They are found in coccus shape. Okay, they are arranged in clusters majority. Yeah, so we call them gram-positive cocci in clusters. Right, this is in example 1. Okay, in example 2, if you look under a light microscope, you see rod-shaped microorganism bacterial cells. They're arranged in chains and they stain pink. Right, remember when cells are stained pink, they are gram-negative. So we call them gram-negative bacilli in chains. Okay, try this now. Look at this image here on the left. What do you see? All right, you should see cocci-shaped bacteria that are stained purple. You can also see rod-shaped bacterium, okay, bacillus-shaped and stained pink. Okay, most of this purple rod-shaped, purple stained cocci, they looks like they are in clusters. Some of them are in chains. Okay, whereas the rod-shaped, bacillus-shaped, pink stained bacteria here, all right, they looks like they are in chains, right? So this should be gram-positive cocci in clusters, and then you have your gram-negative bacilli in chains here. Okay, in summary, we have went through some of the staining principles. We talk about the factors to consider when you prepare specimens for staining and microscopy. Okay, we compare the difference between live and fixed staining, right, as well as positive and negative staining, depending on the dye and the cell components that has been stained, okay, and the objective of your experiment, as well as the microscope that's available for your use. Okay, some of the staining techniques we talk about: simple stains, like using one stain only, like crystal violet, that stains purple throughout all the cells. But if you want to differentiate between gram-positive and gram-negative bacteria, we use a differential stain known as gram staining. Right, gram staining uses crystal violet, iodine, 95% ethanol, as well as safranine, right, for you to differentiate between two different strains. For example, if it's one is gram negative, another one is gram positive. A common gram negative bacteria will be Escherichia coli, right? Another common gram positive bacteria will be your Bacillus subtilis. All right, another stain, differential stain, will be your acid fast stain. Okay, this uses the fusin dye to detect for your mycoplasma tuberculosis, mycobacterium tuberculosis. Sorry. Right, so to detect your Microbacterium tuberculosis, which causes tuberculosis in humans, a lung infection. Right, we also went through the different gram staining procedure. Right, the gram staining procedure involves the different reagent that we talked about just now. Okay, and more details on your practicals. Okay, we talk about the different bacterial shape and arrangement. These can be in cocci shape or in rod shape. They may come in different arrangement, be it in clusters or in singly or pairs, or they may be in chains. All right, so it's important when you describe bacterial cell morphology, take into account their gram-positive as well as their gram-positive status, as well as their shape, as well as their arrangement. So I've included here some recommended reading from the recommended text by Cohen and Smith. So I recommend you to read chapter 3 and 4 here so as to enhance your learning. The information presented here in this lecture notes are minimal. If you were to read this recommended text, I would suggest that you do so as to enhance your learning, and hopefully this will bring your test scores to a higher standing. Alright, so this is the end of part 2 of topic 2. I will see you in the next topic. Thank you.